So today it's a bit of an unusual seminar in the sense that we are not talking purely about computational chemistry or structure-based drug design. I thought that from my role as scientific advisory board member of Biosolve IT, what we all do is we are working on drug discovery projects from small molecules, but also peptides and oligoribonucleotides, perhaps also monoclonal antibodies. And I wanted to summarize a few of the projects that I have seen, and in total three short stories in medicinal chemistry. This is the overview of the three case studies that I'm going to talk about. The first one will be Akynceo Netupitant. As you have seen also in the introductory slide, it is a small molecule that binds neurokinin 1 receptors and antagonizes those. It's today an anti-emetic, FDA approved since 2014, and it's used in oncology to suppress the vomiting and also nausea in cancer chemotherapy. I will then switch gears and talk about a peptide, which is Dasiglucagon, in the treatment of diabetic patients, which is currently in clinical phase 3 trials at Zealand Pharma. And then thirdly, I will be talking, molecules are getting bigger. I will be talking about an siRNA conjugate, conjugated to the N-acetyl galactosamine cluster. That's work from Silence Therapeutics, and it's currently, the clinical trial application has been approved, and this will very soon be dosed into patients in rare blood disorders. Now, Netupitant. We are talking about a highly potent and selective orally active neurokinin 1 receptor antagonist for the treatment of cancer chemotherapy-induced nausea and vomiting. Today, it's used as an oral fixed dose combination with a 5-HT3 receptor antagonist, palonositron, which has been FDA approved, as I said, in 2014. And that's work that we conducted. So I'm going back one slide. That's work we conducted in this building here. So I hope you see my mouse. This is the Discovery Chemistry building at Roche in Basel, Switzerland, at the headquarters, where I started as a chemist, and actually Netupitant, I'm the lead inventor of this molecule, and I later became head of chemistry, and all the chemists are in this building. Now, back to the work at Roche that we conducted. We started with a neurokinin 1 receptor antagonist by simply cloning and expressing the receptor, and we had a parallel screening strategy. So we said neurokinin 1 has to be selective over neurokinin 2 and neurokinin 3 by at least 2 log units, so this is pKis, and we said we wanted to have a molecule that is at least 10 nanomolar, if not better. Then in our parallel screening strategy, we also said, so every molecule that would be fulfilling these criteria, we would test them in cytochrome P450 inhibition, in metabolic stability, and we would front load molecules in to rodent studies in the gerbil, which is in a disease animal model or an animal model in the foot tapping behavior, and molecules that would perform here, we would establish a dose response. And every molecule that would be successful in the second parallel screening strategy would be front-loaded into a third campaign, which would be, we would be testing the molecules for Ames and MNT activity. We would look at a broader panel for selectivity. We would look at a broader panel for cytochrome P450 inhibition. And then again, so you see everything that's in red here is in vivo. We would look in a second species pharmacokinetic behavior of the molecule. Here we selected the dog, and as we are also looking at for an anti-emetic, obviously we would be looking at the ferret, which is an excellent animal model for emesis, and you will see a few data in a second. And then all molecules that have been selected here and would have been positive in terms of their assessment, we would then front load those into parallel toxicology studies, finally for clinical candidate selection. So this was our parallel screening strategy, and that's how we got started and worked on NK1 receptor antagonists. And this is work which is largely 15 years back now, that was conducted at Roche, as I said. So we cloned the receptor. we expressed it in CHO cells, and then we went to our kind of molecule selection, which I would say are the crown jewels of every company, and we screened. In these days, these were only 250,000 molecules, but still we screened 250,000 molecules. And these are the kind of molecular clusters that we received. Yeah, you see polar molecules here. Then here's the benzodiazepine, which is fairly large. Then here, this reminded us of a tricyclic antidepressant, obviously, and so forth. But what really caught our attention was this molecule here. So it originated from a pesticide program. Yeah, so that you can easily see. So 4 chlorine atoms. So not a surprise that this is from a pesticide program. But we really like that molecule, and for the following working hypothesis. So you see here that NK1 receptor antagonists were in literature described, and this here is a model that was derived from site-directed mutagenesis from Chris Swain and colleagues, and that was published. And we actually thought, so if that's true, and there's this magic lipophilic binding pocket here, which contains the bis CF3 substituted aromatic portion. So, and that is a kind of backbone. And here we are having 2 aromatic groups that are edge-to-face interfering here with the histidine 197. So one more time going back, you see here, that's the molecule we found by random screening from the pesticide program. And that's the working hypothesis that we coupled with the screening hit. And that's what we then really selected for chemical optimization. We had good news. The screening hit had a pKa of 7.8, so we were very close to 10 nanomolar. And we had also bad news. There was no in vivo efficacy as tested in the gerbil for the foot tapping behavior. One word to the animal model, you're injecting a neurokinin 1 receptor antagonist into the brain, agonist into the brain, and you're trying to block the response by the orally or IP given molecule, the NK1 receptor antagonist. And you see this in the foot tapping behavior of these animals. Back to the screening hit, we also liked it because it was potent, tractable, small molecules, it's achiral. We were thinking of the cost of goods that shouldn't be too high. It's convergently assembled, so it would lead to rapid diversity, as you will see in a second. It was unique as an NK1 receptor antagonist and as a lead, so it was patentable. We first addressed the linker because the hydrazone, as everyone here in the audience from medicinal chemistry or computational chemistry will immediately see, the hydrazone is not what you really want into to be a part of a medicine. So we went for replacement of the 3 atoms by 5-membered rings. We also had linear chains. We had again methylation, and then we were looking at the amides and also the inverted amides. And what really resulted, I'm cutting this short here. What really was preferred, we found, was the amide, so the inversed amide. It had to be N-methylated, and we also had the amides that are again N-methylated. Here in this case, in the inverted amides, the GEM dimethyl group was really essential for binding. What we've done, we then, this was a crash program at Roche, as I said, 15 years back, when everyone was really working on NK1 receptor antagonists, whether it was Novartis or Pfizer or GSK or MSD, so Lilly, everyone was working on NK1 receptor antagonists, and time to clinical development and time to market was really key for success. What we've done is we worked in 7 chemistry labs in parallel, where I've been leading the chemistry efforts, while there was a pharmacologist, Andrew Slade, who was leading the biology and the pharmacology efforts. And we really, with the best linker, which, as I showed you before, which were either the amides or the inverted amides, with the best linker, we went into a replacement here of the heterocyclic system because we wanted to go into more drug-like behaviors, molecular properties. So we had the isocyclic system, we looked at pyridines, we looked at partially saturated systems, we had isomeric pyridines, another isomeric pyridine series. We started to substitute the pyridine in particular in order to improve the solubility. And in parallel, we also went in 7 chemistry labs, as I said, with more than 20 people in chemistry. We went into the pyrimidine cases, either substituted in this fashion or substituted here on the southern part of the molecule. And I'm cutting a long story short because I'm not showing you more of the structure-activity relationship. This is actually published. So who would like to familiarize yourself with much more of the SAR, you can read this here in Bioorganic and Medicinal Chemistry Letters, as we published this almost 15 years back now. But I wanted to show you is what finally came out is the molecule we are talking about. This is Netupitant, and it's a highly potent and selective orally active neurokinin-1 receptor antagonist. It has at the human neurokinin-1 receptors a pKi of below 1, so it's 0.95 nanomolar, so it's a very active molecule. It was, as I said, also orally active at low doses in 3 animal models. We tested this in the gerbil, which is the animal model for the foot tapping response. Then we tested this in vivo in ferrets, which is an animal model for emesis. And we tested this also in the Sancus murinus, which is a shrew. And those animals are used as animal models in motion sickness. When you're putting these animals onto a rotating platform, they start to vomit. So that's what we tested. And here you see the dose responses overlaid in the ferret, in the gerbil, and in the Sancus murinus. And you are seeing that dose dependently, so 10 milligrams per kilogram, 1 milligram per kilogram, and then 0.1 to 0.01 milligram per kilogram, you're seeing that the animals are completely protected down to 1 milligram per kilogram for the response of NK1 receptor agonism or on emesis, and you're seeing that when we were going below 1 milligram per kilogram down to 0.1 milligram per kilogram, then you're starting to see a breakthrough of the emetic response. And when we are down at 0.01 milligram per kilogram, the animals weren't protected any longer. So these were data that convinced us completely to propose to the board in these days, to the development board, 4 molecules in total. So Netupitant is only one of those, because we had also not only an anti-emetic in mind, we also were looking for antidepressants, what I'm not talking about today, but as the anti-emetic. And the anti-emetic we have been putting into clinical phase 1 trials and have been developing is Netupitant, which, as I said, is the FDA-approved medicine since 2014. We completed the clinical phase 1 studies in healthy volunteers at Roche, where we actually also had a pharmacodynamic marker in healthy volunteers. We injected subcutaneously apomorphine, which is an emetic, and patients were randomized, not patients, but healthy volunteers, apologies, were randomized on placebo or on active on Netupitant, and they received then the apomorphine as an emetic, and we were counting the number of vomits and retchets in healthy volunteers, which was kind of a superimposing of also this dose response, which helped us for dose selection in clinical phase 2. We also conducted in parallel a PET scan, a positron emission tomography scan, to show that we have a sufficient brain receptor occupancy. Now, while this molecule was phase 2 ready at Roche, we decided at Roche to out-license the molecule because peak sales in these days were 850 million. And please remember that in the kind of late 90s, early 2000s, people were looking for blockbuster medicines, and a company like Roche then decided, in order to prioritize clinical development funds, that we would keep royalties but out-license the molecule to Hepcidin, which is a Swiss drug maker, which is privately owned. So we were running the due diligence, and then I'm jumping into clinical phase 3 efficacy studies in patients receiving doxorubicin and cyclophosphamide. So these are cancer patients, and as you're well aware, chemotherapy induces a strong nausea, strong vomiting, and it's so painful for those patients that they either have to reduce the treatment cycle or they have to reduce the dose. And that obviously is then not in line with the treatment cycle that actually from the oncology indication would have been to be used. So we are looking here at a comparison in blue, that was the standard of care. This is palonosetron, which is a 5-HT3 receptor antagonist that's also used for motion sickness in combination with dexamethasone. So that was the standard of care. And you're seeing that roughly 60, 70, 75% of patients would be completely responding to the standard of care. But on the other hand, we still had 30% roughly of patients that still would suffer from nausea and vomiting, where the treatment cycles had to be reduced or the dose had to be reduced. And then this was compared in a clinical outcome trial with NEPA, which is an oral fixed dose combination of Netupitant at a dose of 300 milligram, given together with palonosetron. And that fixed dose combination was tested, as I said, in clinical phase 3 trials in a fairly large number of patients. Here you're seeing the number of patients in the first cycle. You have a certain dropout rate, then the second cycle, the third cycle, and the fourth cycle. And what you can see is that not only the dropout rate was lower, but patients were protected up to 85, 84% against the emetic response. So they were completely protected. And that was due to the working hypothesis that NK1 receptor antagonists would block central NK1 receptors in the nucleus tractus solitarius, which is a brain region where the emetic response in man is funneled through. So this led then finally to Akyncio. As I said, it's a fixed dose combination of Netupitant and palonosetron, which was before we reached the market with this dose combination. That was the standard of care, and that's the new standard of care. In a summary, within 12 months from receptor cloning and expression, we went to clinical candidate selection, and I have to say we've done this with 7 chemistry labs in parallel with more than 20 people in chemistry. The first healthy volunteers were dosed 24 months after project initiation, so 2 years from cloning and expression to first dose in man. So that was kind of a record at Roche. The FDA approved then finally, after the clinical development, Akyncio in 2014 for the treatment of cancer chemotherapy induced nausea and vomiting as an oral fixed dose, as I said, of Netupitant and palonosetron. And also since 4 years, since 2015, this medicine here that you find in all intensive care units for oncology worldwide is now recommended as a preferred medicine by the National Comprehensive Cancer Network. That is an alliance of 26 of the world's leading cancer centers. So, and I really have to say that was a huge team effort. It was fantastic. And for me personally, when I started 20 years back at Roche, that was a learning exercise. This was actually my 3rd project, which I later also had the pleasure to become the project team leader at Roche. And I really thank everyone on the team for that accomplishment, and everyone should be proud that we brought a medicine to patients, which is really a life-saving medicine, because the treatment cycle of cancer chemotherapy can be maintained. I'd like to switch gears, as Markus also indicated. After 16 years at Roche, I decided to join the biotech sector. That was back in 2013, and I became the Chief Scientific Officer of Zealand Pharma. So the company is based in Copenhagen in Denmark, traded on the Nasdaq Stock Exchange in Copenhagen, a public company, and also traded by now on the Nasdaq Stock Exchange in New York. A very successful company, which is purely based on peptide-based medicines. And I'm talking about a peptide here. The molecules, as you noticed, are getting a bit larger. We were looking at a stabilized and soluble glucagon analog as a liquid dosage form. And what we had in mind is a ready-to-use rescue pen for the treatment of diabetic patients with severe hypoglycemia. And the second project phase would be that the molecule becomes a component of a dual hormone artificial pancreatic system that diabetic patients would be wearing at the belt, and it's becoming a reality. This molecule that I'm talking about is now in clinical phase 3 trials. It will be submitted as a new drug application probably in 2020, and there are already very positive clinical phase 3 results that have been published by Zealand Pharma. Now, why were we interested in a liquid dosage form of glucagon. So first of all, this here is a real case. This is a patient that is dead by now. So unfortunately, this patient was wearing a glucose, an artificial insulin pump at the belt. And this patient, so we're seeing here the glucose levels in milligrams per deciliter, how it's typically reported as clinical parameter. Here you're seeing there were a few correction boluses of the insulin pump given based on subcutaneous measurement of glucose. So whenever the critical level of glucose would be reached, this is obviously a type 1 diabetic patient, the insulin pump at the belt would be releasing insulin and would give a bolus in order to correct for glucose levels that were measured subcutaneously in an open loop system where insulin is injected automatically and glucose is measured subcutaneously with a glucose sensor in this patient. Now this patient also did exercise, meal, and had twice food, and there was an overestimation by the pump of insulin injection. So we're seeing here insulin injection, which was then fatal because the patient went to bed, went into hypoglycemia, went into the subcritical level of where the consciousness is lost. And this patient, so there were two possible glycemic responses to counter-regulatory hormones, and this patient was found dead in bed in the morning. So, and these data, this is not unique to that single patient. So that's a real threat for type 1 diabetics. And we thought we need a rescue pen. And why do we need a rescue pen? This is because in the case I've showed you, this was a patient that went to bed, but in typical hypoglycemic events are occurring in type 1 and type 2 diabetic patients very often. And blood glucose, when blood glucose goes down, so what patients, as long as they are conscious, can do, they typically drink orange juice, or they eat some bread, or take the direct glucose. What happens if we are going below the 70 nanogram level per deciliter, then they are going into the unconscious mode. They are typically then not responsive any longer. And what is on the market, still is on the market, and was on the market when we initiated the program, is a rescue pen. But the rescue pen, and I'll show you in a second, has a big drawback. Native glucagon is not stable as a liquid dosage form at room temperature, so it crystallizes out. That means one has to reconstitute the material. So the rescue pen that you find on the market, either from Lilly or Novo Nordisk, you're finding that there is a syringe which is pre-filled with saline, buffered saline, and then you're finding here a vial with lyophilized glucagon. So just imagine you have a patient on the floor, for example, it would be your child, a type 1 diabetic child, unconscious, and you would have to carefully reconstitute before injecting this subcutaneously, and you have to agitate this for 30 seconds. You are not allowed to shake it because then you're having bubbles. So what is happening? So people have this at home. In 95% of the cases, it is not used. What they do is they call emergency 911. So this is the emergency call in the US. So that's what patients currently do. And that was the kind of background of our working hypothesis. And as I said, this is the current treatment. You have the patient unconscious, and then you has to start reading the manual, how you're reconstituting the material. And that's, as I said, that's why the reconstitution kit is, everyone has it at home, but it's not used. And this led us to our project where we said we'd like to have a ready-to-use rescue pen with liquid glucagon as a formulation, and that was the working hypothesis that we had back in 2013 when the molecule was pitched to the board of Zealand Pharma, and it was actually accepted as a development program, which is now in clinical phase 3, as I said in the outset. So the first generation would be, we are now jumping to the pump, as I said. So the rescue pen was phase 1 of our project, and we also looked in parallel at a potential development as glucagon for a dual hormone pump. You've seen the patient that died overnight was wearing a glucose pump like this with subcutaneous glucose measuring, and there was an overdose that drove this patient into hypoglycemia, and in the end, this patient even died. This is not uncommon. that the glucose, that the insulin pump is overdosing, you have to compare this. You're running a car here, the insulin pump, you're running a car with no brakes. So that's the comparison of the first generation. Still, 75 to 80% of all diabetes 1 patients in the US are wearing such an insulin pump. And we said that if this is the first generation, what about the second generation? It would be a dual hormone open loop, a continuous glucose monitoring with both insulin and a glucagon pump. So you would have a dual chamber pump. That was our kind of vision that we had at this point in time. And it is, as we speak, currently in developing a clinical phase 3 development by Zealand Pharma in collaboration with big pharma partners. And there would be then in the second generation a dynamic release of insulin and glucagon. And if this patient that went into the dead in bed syndrome that I showed you on a few slides ago would have been on such a pump, obviously glucagon as a liquid dosage form would have been injected to counterbalance the insulin overdose. We had a target product profile. For the rescue pen, we said it has to be stable for 1 year at 5 degrees Celsius, followed by another year at 25 degrees Celsius in liquid formulation. So people would buy this, would store this in the fridge, and then they have one pen in use, which would be valid, quote unquote, for 1 year. Then has to be replaced because the material also starts to degrade in a liquid dosage form. We are looking obviously at a peptide, which is glucagon. Then we also said the pharmacodynamic and the pharmacokinetic profile has to be comparable to native glucagon, which has to be as close as possible. It has to be safe and well tolerated. In the second arm of the project, our target profile was it has to be stable over 1 year at 5 degrees Celsius, followed by 1 week at 37 degrees Celsius, because this dual hormone pump is worn at the belt, and therefore we said let's go for the extreme, for the temperature for the body temperature, 37 degrees Celsius. Again, PD, PK has to be comparable to native glucagon. Obviously, it has to be safe and well tolerated. I'm not going into details, as I also didn't go into Netupitant in the entire SIR, but a number of molecules have been designed and synthesized and tested. Obviously, there has been a very strong rationale. If you'd like to read more about this, we published in Drug Discovery Today a general peptide therapeutics review article, current status and future directions, which has been published, as you can see, in 2015, and that's one of the graphs that I've taken out there. Obviously, how you're doing peptide design. It's initiated often by an ELA scan, followed by a synthesis of focused libraries. Once one has understood the functions of the individual peptides, amino acids, then further substitution drives the structure-activity relationship, leads to selection, synthesis, and then the EC50 determination. This is how we went from an endogenous peptide, glucagon, to new peptide ligands. And as I said, so glucagon crashes out as a liquid dosage form in room temperature and cannot be stored. We were looking with a focus on stability and formulation for a liquid dosage form. And glucagon is a 29 amino acid peptide sequence. And here the amino acid sequence of dasiglucagon is shown here. And you're seeing in blue the critical amino acid replacements that have been conducted by the chemists at Zealand Pharma, a very strong team. And so Ditte Rieber is actually the scientist who designed this molecule and synthesized it for the first time. Here we are seeing animal data in anesthetized rats. So we compared, obviously, native glucagon, which is then here, that's the blood glucose. We compared glucagon at 20 nanomoles with dasiglucagon, where you're seeing there is already at 6 nanomoles per kilogram, you're having a comparable response. And then when we are going to this highest dose, the 20 nanomoles per kilogram, you're having even a superior profile. So this is a single subcutaneous injection in anesthetized rats, and you're seeing increased bioavailability. And then the in vivo potency is similar with a fast pharmacodynamic onset. So these kind of data convinced us to present the molecule to the board. We also had then a rapid rescue from insulin-induced hypoglycemia in rats. You're seeing this here. Again, we are comparing glucagon at 20 nanomoles per kilogram to Dasiglucagon, compared then also to vehicle. And you're seeing when at time point 0 we dosed insulin. So remember, that's the kind of animal model for a patient who is on an insulin pump. So we are having here the insulin injection, and then you are going into blood glucose, where this is then lowered, and we, with glucagon or Dasiglucagon, rescued. After 45 minutes, we rescued the animals, and as you can see, in a very beautiful parallel behavior, the pharmacodynamic effect was observed in rats, and as a conclusion, Dasiglucagon and glucagon demonstrates similar pharmacodynamic effects in rats. As I said, so these data convinced us really to go to the board and present this for a clinical development program. One more word to the chemical stability. That was obviously also one key for proposing the molecule. We had a target profile where we said, these are the stop or go criteria, and these are the 3 months chemical stability data at 40 degrees Celsius, accelerated stress test conditions, 25 degrees Celsius and 5 degrees Celsius, how this product, the medicine, would be stored by patients in the fridge. And as you can see, after 3 months, there was a quantitative molecules were left, and also more than 90%, so up to almost 98% of material was recovered when stored for 3 months at room temperature. So these were sufficient parameters, and here you're seeing again glucagon. So these are the fibers, the amyloid-like glucagon fibrils, how this crashes out, and that this is just native glucagon is not cannot be provided in a liquid dosage form. So in summary, Dasiglucagon, and here on the right-hand side, you are seeing the clinical development and preclinical development pipeline of Zealand Pharma. You're seeing here Dasiglucagon as the rescue pen, Dasiglucagon in a rare disease, and also Dasiglucagon in the dual hormone pump. So this is taken today, this morning, from the web page of Zealand Pharma. You are seeing this program is in clinical phase 3 development. Hypopal, in summary, 99% of subjects in clinical trials were recovered from insulin-induced hypoglycemia within 15 minutes. As I said, the molecule is currently in phase 3, has also pediatric data generated that were published this week, also positive on all primary and secondary endpoints. There's a collaboration with Beta Bionics in the development of the dual hormone pump, so the artificial pancreas is currently in phase 2, and it's in preparation for clinical phase 3. And then it's also in a CHI, a rare disease affecting newborns that are caused by a defect in pancreatic cells resulting in insulin overproduction. This is also in clinical phase 3 currently. The FDA and the European Medical Agency granted orphan drug designation to Dasiglucagon, and so for this rare disease. Again, as I did for Netupitant at Roche, for Dasiglucagon at Zealand Pharma, I can only congratulate the team for a fantastic work, and for a super energetic teamwork that has brought this molecule within a year into clinical development, from having it been synthesized a year into clinical development, and now with very rapid progress now in clinical phase 3. And here's the peptide sequence again. I'd like to switch gears. Molecules are getting, again, bigger. This is SLN124, a N-acetylgalactosamine siRNA conjugate for the treatment of rare iron overload disorders. And this is work that I've been leading at Silence Therapeutics, headquartered in London, with laboratories here in Berlin. And again, this is a public company traded on the Nasdaq, not the Nasdaq, the London Stock Exchange, on the alternative investment market. And I joined this company first as Chief Operating Officer, later as Chief Scientific Officer. And this is an RNA company, and we focused, with my arrival, we focused completely of siRNA and acetylgalactosamine conjugates for hepatic delivery. Why did we focus on this? When I was working at Roche, I was leading also the acquisition and the oligoribonucleotide delivery work of Alnylam Europe that became part of Roche, and we had been working on siRNA GalNAc conjugates, and we knew before divesting siRNAs again at Roche that this technology works. So that's why with my arrival at Silence Therapeutics, we focused on that. That's just a schematic representation. siRNA molecules, this is taken from Klostermann in biochemistry. These are fairly large molecules. Yeah? So 2.5 helices typically for a 21 mer. You have a molecular weight of 12 to 14 kilodalton. This is as long as a phospholipid bilayer is thick, so 55 angstrom, 20 angstrom here in the diameter. It has 40 negative charges. It's heavily hydrated as it depicted here, and a molecule like this will not passively diffuse over a membrane. So it's just a no-go because of the negatively charged phospholipids that are found in the cell membrane. But what obviously was known and that we used and extensively also published and patented, if you couple the siRNA gene silencing sequence through a linker to the N-acetylgalactosamine hepatocyte targeting ligand, then obviously those can be internalized through receptor endocytosis going into a lysosomal release intracellularly. And here you're seeing again, so this is the chemical modification stabilization pattern. The RNA is degraded, so it has to be stabilized, mediates gene silencing. An antisense strand is coupled with the sense strand, and to the passenger strand, we, through a certain linker, couple then here the N-acetylgalactosamine. Here you're seeing the cluster, which is known to bind to the receptors on hepatocytes. And as I said, you're seeing this here. This is the extracellular compartment. This is the intracellular compartment. The N-acetylgalactosamine siRNA conjugate binds to certain receptors, the ASGPR on hepatocytes. It's then engulfed the receptor upon activation. It's internalized. It ends up into an endosome, which then undergoes a pH drop into a lysosomal release. The receptor is recycled to the membrane, while the siRNA becomes available for gene silencing as the RISC complex, which then finally leads to messenger RNA silencing. And we use this for the treatment of iron overload disorders. Here we had the project rationale selected a hepatic target, which is a membrane-bound enzyme, which is negatively coupled to hepcidin, which again is negatively coupled to iron and iron uptake in serum. And here you're seeing animal data in mice. We are injecting on day 1, and on day 4 we are observing then dose-dependently 1, 3, 10 milligrams per kilogram. We are observing dose-dependently the messenger RNA knockdown in animals, in mice. Then very nice, beautifully, we are seeing here the network pharmacology. Hepcidin is negatively coupled to Timpris. This was known in literature. And as we anticipated, there's a dose-dependent increase of hepcidin. And then again, after 4 days and a single administration in mice, we are seeing that iron levels go down. And this is actually the central disease burden of patients with iron overload disorders. So we have proven the mechanism, and we move then into a 6-week studies where we dosed on day 1, and after 1 week, 3 weeks, and 6 weeks, we were looking for the pharmacodynamic effect. So remember, it's a single dose, and we observed up to 6 weeks the animals. What you see after 1 week, 3 weeks, and after 6 weeks is you're seeing the messenger RNA of the hepatic target is knocked down. It starts to come back after 6 weeks, but there's a long duration here, a long duration of action in mice. And here you're seeing that also the iron levels in week 1, week 3 are sustainably reduced, and after 6 weeks they start to come back, which obviously is in line with what we have seen at the mRNA level. We then looked also at the efficacy after a single dose administration in non-human primates. We injected NHPs on day 1, observed them up to 10 weeks. So remember, this is these are very, very strong efficacy data. A single subcutaneous dose in cynomolgus monkeys led then to a sustained reduction. So we're looking here at 10 milligrams per kilogram, 1 milligram per kilogram compared to vehicle. We start to see the messenger, the circulating iron levels, they start to come back after weeks after the single dose administration, and they are back after 10 weeks. So well tolerated with a long duration of action. Again, this is what convinced us to pitch the molecule to the board of directors at Silence Therapeutics. The CTA was filed this year, and the first patient dosed is to be expected in 2019. Obviously, since I'm now at Taros as Senior Vice President for Drug Discovery, this is work that was conducted in these days in London and Berlin. Fantastic work, and I can only again congratulate the team and the company for the accomplishment. The European Medical Agency granted orphan drug designation for SLN in the treatment of beta thalassemia, which is a rare disorder for iron overload. The start of clinical phase 1b trials with beta thalassemia, and also the myelodysplastic syndrome is expected, as I said, in the second half of this year. And the primary endpoint will be safety and tolerability in patients with efficacy markers as a secondary clinical endpoint. I don't want to let you go without saying a few words about future trends in medicinal chemistry. You've seen a small molecule, you've seen a peptide, and you've seen an oligoribonucleotide conjugate. And I think as we move on in medicinal chemistry and as we broaden our horizon, we are witnessing, and we will witness in future, a broad maturation to chemical biology, where we are using phenotypic screens, for example, in insulin-resistant pancreatic beta cells. And as we speak at Taros, I'm running a project together with the German Diabetes Center, where we are using exactly that resistant pancreatic beta cells, and we are looking for molecules that are secreting insulin again, and we found a lead which also works in vivo. Then I'm very convinced that a structure effect and structure activity relationship-based knowledge management for pattern recognition will be seen more and more and more, and the chemical space navigation that we published, Markus and myself, from BioSolve IT together with Taros. It's just an example of how we are advancing the entire field of what is called today, I think, artificial intelligence. Then I'm convinced that intracellular delivery of polar macromolecules, we will learn more and more and more. We have the cell-penetrating peptides. We have the examples of siRNA conjugates delivered through endocytosis. And I'm convinced that since we are understanding molecular modules and their function, like the N-acetylgalactosamine, like the linkers, like the siRNAs, this can be coupled to a peptide, for example. Antibody drug conjugates are in clinical development. So this will be a future also for medicinal chemists to explore larger molecules. Regenerative medicine approaches, there are mechanisms for cellular self-renewal. It's working endogenously in animals. Why shouldn't that work in man? So, and there's a lot of work conducted on induced pluripotent stem cells from somatic cells and redirection of cell fate. As we speak at Taros, we are running a project together with Z Clinics in Spain, Z Cardio, where we are looking for cardiocyte regeneration because we are using zebrafish. Zebrafish are known that injured cardiomyocytes, they are recovered by an endogenous mechanism in the larvae, and that mechanism is still present in man. It's just not executed, and we are looking for molecules that can rescue cardiomyocytes. And last but not least, RNA as a drug target, small molecules that regulate gene expression. It's very clear to me that if we are targeting the messenger RNA with a small molecule, which binds to the 5' end of the untranslated region, that molecule would then prevent the messenger RNA of a protein which is causal for disease. The messenger RNA would not be read at the ribosome because it's blocked by a small molecule that binds to the 5' end of the untranslated region. And this way, we would be silencing a gene not at the gene level, but at the translational level, at the messenger RNA level. And I'm convinced we will, in a few years from now, we will have molecules in clinical development where RNA is the primary drug target. I'm closing here. That's my last slide. If you have any questions, contact me here on this email or on LinkedIn, or send an email to Markus, or now use the chat for placing questions. I also would like to mention there's a strong colleague of mine at Taros a senior medicinal chemistry expert, Torsten Gensky. So you figured that you have to be called Torsten to work at Taros in drug discovery. No, joking aside. So he is also, we two are here running the drug discovery and medicinal chemistry leadership together with computational chemistry. And I stop here and take any questions. Thank you for your attention, and let's see whether there are questions. Thank you.